Imagine a diploid cell during interphase. The chromosomes are long, thin strands of DNA within the nuclear envelope. There is a single pair of centrioles near the nucleus. Near the end of interphase, the cell prepares for the first phase of meiosis, meiosis I. The centriole pair duplicates. Each chromosome is replicated. Homologous chromosomes begin to group together. Meiosis I begins. The first phase of meiosis I is prophase I. During prophase I, the centriole pairs move to opposite sides of the cell. The chromosomes condense. The homologous chromosomes are tangled together. A total of 4 chromatids are present, since each chromosome was replicated before meiosis began. Each pair of sister chromatids is joined at a centromere. While the pairs of chromosomes are close together, they may break and exchange segments. This is called crossing over. The nuclear envelope dissolves and the spindle fibers form. The homologous chromosomes remain paired as spindle fibers pull them toward the center of the cell. The second phase of meiosis I is metaphase I. During metaphase I, the homologous chromosomes line up in the center of the spindle. The third phase of meiosis I is anaphase I. During anaphase I, the homologous chromosomes are pulled apart by the spindle fibers. They move to opposite ends of the cell. The fourth phase of meiosis I is telophase I. In some cells, the nuclear envelope forms around the chromosome sets. Cytokinesis begins when the cell membrane starts to pinch the cell's cytoplasm in half. 2 new daughter cells are formed. Each daughter cell is haploid. Each has half the chromosomes from the parent cell. In preparation for another cell division, the centriole pairs duplicate. The chromosomes do not duplicate. Each daughter cell is now ready for meiosis II, the second cell division of meiosis. Meiosis II is very similar to mitosis. However, meiosis II starts with a haploid cell. The first phase of meiosis II is prophase II. During prophase II, the centriole pairs move to opposite ends of the cell. The nuclear envelope dissolves and a spindle forms. The chromosomes are pulled toward the middle of the cell. During metaphase II, the second phase of meiosis II, the chromosomes line up in the center of the spindle. In anaphase II, the centromeres of the sister chromatids separate. The chromatids are moved to opposite ends of the cell. Each sister chromatid is now considered an individual daughter chromosome. The last phase of meiosis II is telophase II. During telophase II, nuclear envelopes form around the chromosomes. In cytokinesis, the cell membrane pinches the cell in half. Each haploid cell has now divided into 2 haploid daughter cells. The original diploid cell has given rise to 4 daughter cells, each with a single set of unduplicated chromosomes.